Hello students. Now today we are going to read mutation, genetic code, then translation and human lac operon model, and then after human genome project also, right? Mutation here, mutation already we read in our previous lecture. So here only two type of mutation is given here, only frame shift mutation and point mutation we will read here, right? And different type of mutations it is already read by us in the previous lecture. Okay, so now we start here. Mutation, simply I am asking for you, what is mutation? Mutation means what? A sudden change in the genome which is transferred into the next generation, right? Means we can say sudden changes which can transfer from generation to generation, they are called as mutation. Mutation means it is actually altered form or it is changing in DNA fragments, right? Okay, the relationship between the genes and DNA are best understood by the mutation studies. Mutation, who gives this mutation theory? We very well know, that is Hugo de Vries, right? He gives us. You have studied about mutation and its effect in chapter 4, means in previous chapter already we read the different type of mutation and its effect. Effects of large deletion and rearrangement in a segment of DNA are easy to comprehend. We understand how a mutation occurs, means large deletion occurs or rearrangement, right? It may result in loss or gain of a gene, and so because loss of gene means loss of function, gain of a gene means additional function is there, and it may be harmful or beneficial to the new generation, right? The effect of point mutation will be explained here. Point mutation, here what is given? The effect of point mutation. Here only point mutation is given, will be explained here. And you know point means what? A simple point means a nucleotide. Single nucleotide is, that means it is split off or it is exchanged with some another, or it is replaced by some other nucleotides, maybe we can say, right? A classical example of point mutation is a change of single base pair. That is, single base pair means what? Single nucleotide in the gene for beta globin chain. Now we are talking about the sickle cell anemia, okay? Because here it is given a single base pair in the gene for beta globin chain, right? That results in the changing of amino acid residues glutamate to valine. Already we read this one in our previous lecture, right? That means which this glutamate is changed, right, to valine. It results into a disease condition that is called sickle cell anemia. Effect of point mutation that insert or deletes a base in structural gene can be better understood by the following simple example which is given here, right? Here only they explain how the genetic code altered, right? Because genetic code alter means we know very well different genetic code for different, they are responsible for the different amino acids. That means what? If a genetic code is altered, right, then what happens? The amino acid also. Amino acid is altered, that means what? The protein is altered, right? That's why here sickle cell anemia is formed. Consider a statement that is made up of the following word, each having 3 letters like genetic code. This Ram, 3 letters is there like genetic code, has red cap. We know, understand? Okay, we understand Ram has red cap. We can. If we insert a letter B in between has, suppose that in between has and red, if we insert a letter B, then what happens? It would read as follows: Ram has, because we know genetic code means it is made up of only 3, only 3 base pair, right? It is given here. Ram has B, because we add B, so B replace the other. D come to the C. D, this D, it comes to the capital. So B, D, C, A, P, the total meaning is changed. This one is frame shift. This one is also a point mutation, but it is frame shift. Frame shift means what? If we add one more, then means what happens? This one is changed, that one is changed. We can say after has, red is changed, cap is changed. If other nucleotides are also, if other genetic codes are there, all the genetic codes they are changed. So that means we can say here total frame is changed. That's why it is called as frame shift mutation. Ram has B, D, C, A, P, right? Similarly, if we now insert two letters, suppose that B is added, then it is changed. Suppose that we have insert one more, that is I. Then what happens? Ram has B, I, R, E, D, C, A, P. That means that remaining genetic codes are changed. Or we say the total frame is shifted to left side, right? If one or two letters are inserted, that means what happened? Inserted, right? It is not a deletion, it is not a replacement, right? It is not exchanged, but it is inserted. Then what happens? The genetic code from that region, it is totally shifted, right? Means all the genetic code, they are different. Different means all the amino acids, they are different. A different type of protein is formed. But now we insert three letters together, say big. The statement would be read as Ram has big red cap. Now here you understand, here Ram has same genetic code is there, red cap, it is the same genetic code which is present in our original or mother chromatin thread, but one extra genetic code is added here. That means what? One more amino acid is added. This one is really point mutation, right? The same exercise can be repeated by deleting the letter R, E, add one by one and rearranging the statement to make the triplet word. This one is additional. If one or two is added, means total frame shift is there, means all the amino acids are changed. But if we add 3, that means what happened here? Only one amino acid is changed, means one more amino acid is added here. That is called addition, insertion. So this is the example of insertion. Now deletion means if we delete one word, like here Ram has, now here red R, we delete the R, then what happens? It becomes EDCAP. That means what? If we delete, that means also it is shifted toward the right side, right? That means from this region, all the genetic code is changed. All the genetic code change is what? All the amino acids are changed. Here Ram has two E and R, both are means delete. Then what happens? Ram has D, C and P. Same process. Means here after this has, what happens? All the genetic code is altered. All the genetic code, right? It is altered. That means what? All the amino acids are altered. Now, if 3 is deleted, then one whole, that is what? Which one? Ram has red, right? So red is totally deleted. Ram has cap. That means what happens in new protein? What happens? One amino acid is deleted. Do you understand here? This one is a point mutation, but all the other amino acids, they are same. Only one amino acid change. But here what happens? In these two cases, all the amino acids after this region had changed. But here only one amino acid is added. Here what happens? One amino acid delete. This one is called deletion. But here what happens? In the same process, means all the amino acids are altered, but here what happens? One amino acid is added, that is called addition, right? Addition and deletion, point mutation, right? Do you understand what is frame shift and what is point mutation? Frame shift means what? If we remove, right, or we add one or two nitrogen base, or we can say nucleotide, then what happens? All the genetic codes after this region are altered, right? This one is called as frame shift mutation, right? But if we add or delete 3 nitrogen base or nucleotide, then what happens? Only one amino acid is deleted or it may be added. If it is deleted, this one is called deletion. If it is added, it is called addition, right? If 3 add or 3 delete, this one is point mutation, and if 1 or 2, then they are called as frame shift mutation, right? Do you understand? If we add or delete 1 or 2 nitrogen base or nucleotide, then they means the total frame is shifted. That means what? All the amino acids are altered due to the alteration of all the genetic codes. This one is called frame shift mutation. And if we add 3 or we delete 3 or in multiple of 3 or multiple of 3 means deleted, then it is called as frame shift mutation. Do you understand? The conclusion for the above exercise is very obvious, very clear. Insertion or deletion of one or two base change the reading frame from the point of insertion or deletion. However, such mutations are referred to as frame shift insertion. Frame shift insertion, if we insert, it is called frame shift insertion, and if we delete, it is called frame shift deletion, right? 1 or 2, right? And insertion or deletion of 3 or its multiple bases insert or delete in one or multiple codon, hence one or multiple amino acid, and reading frame remains unaltered from that point onward, so it is called as point mutation. You understand? Point mutation means two types. Point mutation one is, and another one is frame shift mutation. Okay. Other mutations already we read, so I don't think that we need to hear once more. Right? Now, next point is our transfer RNA, the adapter molecule. This transfer RNA already I think we read about this transfer RNA. This is also called as adapter RNA, or it is also called as sRNA because it is soluble, right? So it is also called as, earlier it is named sRNA, adapter RNA or transfer RNA, right? This is the smallest RNA because it is made up of 4 sRNA, right? From the very beginning of the proposition of code, it was clear to Francis Crick that there has to be mechanism to read the code and also to link to the amino acid, because amino acids have no structural specialties to read the code, right, uniquely. He postulated the presence of an adapter molecule that would on one hand read the code and on other hand would bind the specific amino acid. He thinks it means it has two sides. One side to recognize or identify or read the codes, and other side means another side where specific amino acids are attached, right? The transfer RNA then called at that time, it is called sRNA, that is soluble RNA, was known before the genetic code was postulated. At that time it is called sRNA. However, its role as an adapter molecule was assigned much later, right? Transfer RNA has an anticodon loop that has base complementary. Anticodon because codon is present in messenger RNA and it is just a complement to the codon. As for example, suppose that a messenger RNA in which code is AUG, that is, you know very well, this time it is a initiation codon and which is response for methionine, right? AUG is there in messenger RNA, that means what? In transfer RNA what is there? We can say transfer RNA is just complement to this one, right? UAC we can say, U, like here UAC is given, that means what? Transfer RNA is AUG, U just complement, this one is complement. So this is codon, that just complement of anticodon. Transfer RNA has an anticodon loop that has bases complementary to the code, and it also has an amino acid acceptor and it has 4 arms, right? According to this cloverleaf model. Cloverleaf model, it is given by Hawley, right? It has 4 arms. One enzymatic arm is there, one attachment arm is there, one anticode arm is there, and one amino acid acceptor arm is there, right? That is to which it binds the amino acid, specific amino acid, because we have 20 amino acids, so we can understand that we have 61 transfer RNAs, but they are specific for 21 amino acids, right? For 20 amino acids means 20 different type of enzymes are present, means these are specific for enzymes, specific for amino acids also. Transfer RNA are specific for each amino acids. For initiation, there is another specific transfer RNA that is referred to as initiator transfer RNA. Initiator transfer RNA. There are no transfer RNA for codons. means 61 transfer RNA are there, but we have how many codes are there? We have 64 codes are there, but we have only 61 transfer RNA because for the code UGA, UAG and UAA, there is no one transfer RNA. Second, there are no transfer for stop codon. The secondary structure of transfer RNA has depicted. Secondary structure and tertiary structure already you know. Primary structure means what? It is a linear structure, right? Secondary structure means what? Surface structure is there. Means here only length and width is given. Tertiary structure means what? Tertiary means three-dimensional structure we can say, right? It has length, width and height is also there, right? So here the secondary structure of transfer RNA has been depicted that looks like a cloverleaf model, that is called cloverleaf, right? So cloverleaf, that is also called, it is given by Hawley, right? And actual structure, the transfer RNA is compact molecule which is inverted L, like to inverted L. Actual structure is this one, adapter molecule, right? Hawley, and this structure it is given by Kim. Two scientists are there, right? This cloverleaf model it is given by Hawley, and this inverted L it is given by it is given by Kim, right? It is three-dimensional structure. This one is three-dimensional. Structure, and this one is two-dimensional structure. That is the difference between these two. Okay. And this structure of transfer RNA already I tell you in this chapter is also, but in the previous lecture we already discussed about the structure of transfer RNA deeply, right? So this time we need not to discuss once more. Okay. Now we start direct from translation. Translation means what? Translation means formation of protein. Formation of protein, simply we can say. So translation refers to the process of polymerization of amino acids. What is protein? We can say polymer of amino acids are called protein, right? And polymer of amino acid, what is the process in which amino acid is formed? It is called polymerization of amino acids. So this is given here. Translation refers to the process of polymerization of amino acids to form a polypeptide. Polypeptide. Peptide means what? A bond between two amino acids. Means a bond between NH2 amino group and COOH group of amino acids, means carboxylic group and amino group. This bond is called peptide bond. And already we know if it is a carbohydrate means glycosidic bond, protein means peptide bond, if the fat means ester bond is there. Okay. The order and sequence of amino acids are defined by the sequence of bases, right, in the messenger RNA. Because messenger RNA it has code. So sequence of bases means triplet sequence of bases. They define the amino acids. The amino acids are joined by a bond which is known as peptide bond. Right? In previous class in 11th, I tell you about the structure of protein. Right? How a peptide bond is formed. You can see here. I can write here. This one is our COOH, right? And this side R, L, and that one is H. Variable R is a variable group. Here NH2 is there, right? This side another amino acid is there, and H2. This side C, R, H, C, double OH, right? We can write here C, double O, OH. Okay, here C. Here also I write this one, right? Otherwise you will so this, okay, you can understand here. This is also the structure like this one, right? So you understand here this OH and OH. H2O is removed here. H2O is removed, right? Now a structure between C and N. Structure between this C is here. Right? So C and N. This bond is called, here you can understand. Here N, H, H, C, R, H, C, double bond OH. So when you see this one, you understand, OH, this H2O is removed. Now a bond is formed between these two. This bond, this one is called peptide bond, right? Between C and N, carbon of carboxylic group and the nitrogen of amino acid, that is amino group. A structure is formed, and this structure, this bond is called peptide bond, right? Peptide bond. Do you understand here? The amino acids are joined by a bond which is known as peptide bond, and peptide bond, it is formed between carbon and nitrogen. Carbon of carboxylic group of one amino acid, it binds with nitrogen of amino group of another amino acids, and this bond is called as peptide bond. Okay. Therefore, in the first phase itself, amino acids are activated in the presence of ATP. So first process here. Okay. What happens? Amino acid, it is attached. Amino, because without activation, without energy, no process happens here. So amino acid, by using one at ATP, it becomes activated. That is called amino acylation of transfer RNA or activation of transfer RNA. That is activated and linked to the cognate transfer RNA. A process commonly called as charging of transfer RNA. Here only in one step it is given. First, by using ATP, amino acid is activated and then it attached with transfer RNA, specific transfer RNA, that is called charging of transfer RNA or amino acylation of transfer RNA. Right? Amino acylation means amino acid attachment of with transfer RNA, that is to become a more specific. If two such charged transfer RNA are brought close enough, then what happens? The formation of peptide bond between them. Right? If these two transfer RNA, they come too close to each other, then what happens? Automatically a peptide bond is formed between these two amino acids, right? That is given here. Would be favored energetically, but it need energy, and this energy comes from GTP, right? Magnesium is also helpful here. The presence of catalyst would enhance the rate of peptide bond formation. Which catalyst means which enzyme is used here? Ribozyme. Ribozyme means what? If it is a prokaryote, it is 23S RNA, and if it is eukaryote, then it is a 28S RNA. These are called as ribozyme. The cellular factory responsible for the synthesis of protein is the ribosome. Ribosomes are also called as protein factories, right? Ribosomes because they perform the platform where protein synthesis occurs. So it is called the factory. The ribosomes consist of structural RNA and about 80 different type of proteins. 80 proteins and specific structural RNA, and already we know the structure of ribosomes, right? When I tell you the ribosomal RNA, at that time I tell you a ribosome, either it is 70S or 80S, they are made up of 2 units, and these 2 units are also made up of some specific RNA, right? In its inactive state, it exists as 2 subunits, one large subunit and a small subunit. When the small subunit encounters an mRNA, first what happens? This small subunit, it attaches with messenger RNA, right? That is given here. When the small subunit encounters an messenger RNA, the process of translation of messenger RNA to protein begins, right? There are 2 sites in the large subunit, right, for subsequent amino acid to bind to and thus be close enough to each other for the formation of peptide bond. The ribosomes also act as catalyst, as an enzyme, as it is given here. In case of prokaryote cells, 23S RNA in bacteria. Bacteria means prokaryotes, but in eukaryotes means it is 28 rRNA, right? Is the enzyme ribozyme for the formation of peptide bond. Okay. A translational unit in messenger RNA is the sequence of RNA that is flanked by the start codon. Start codon means it is AUG, right? Sometimes what happens, GUC also initiate the translation process, but our chief initial codon is AUG, methionine, right? And the stop codon and the code for polypeptides and the stop codon means what? We know very well, UGA, UAG and UAA. And mRNA also has some additional sequences that are not translated. They are present before the before the 5' end, that is initiation codon, and after the terminating codon, that is at the 3' end. And they are also called as, because they are not involved in translation, so they are referred to as untranslated region, or in shortcut we said them UTRs. UT means what? Untranslated art region. The UTRs are present at both 5' end before, right, start codon, and 3' end after terminating codon or stop codon. They are required for efficient translation process, right? They cannot translate, but they are required for efficient translation process. For initiation, ribosome binds to the messenger RNA at the start codon AUG, that is recognized only by the initiated transfer RNA, right? Initiated transfer RNA means what? That is the code. mRNA, the start codon is AUG, that means initiate transfer RNA. You know complementary anticodons are present. So what is the complementary anticodon of AUG? It is, we can say, we understand A means what in RNA? It is U, and U means what? A, and G means C. UAC, right? The ribosome proceeded elongation phase of protein synthesis, and during this stage, complexes composed of amino acids linked to transfer RNA sequentially bind to the appropriate codon in messenger RNA by forming complementary base pairs with the transfer RNA anticodes, right? The ribosome moves from code to code, right? You can say codon to codon, right, along the messenger RNA. Amino acid, this process is called, when ribosome is moving on messenger RNA, so this process is called translocation. Okay, translocation. Amino acids are added one by one, translated into polypeptide sequences dictated by DNA, right, and represented by the messenger RNA. At the end, a release factor binds. A release factor binds to the stop codon, terminating the translation and releasing the complete polypeptide form from the ribosome. That is the total process of translation, right? Now here, we can understand very easily how a translation process occurs here. When we consider the translation process, the translation process means what? It in translation, process, what happens? In translation process, we have 3 steps. How many steps are there? 3 steps. Which steps? First step is our initiation, means start, right? First, because before initiation, what happens? Activation. In our NCERT is also given activation of amino acids. So first we can see here activation of amino acid. How a amino acid is activated? In NCERT is given by using of 1-ATP, right? Yes, that is correct. That means we can say here activation of amino acid, what happens? Here amino acid plus ATP, because without enzyme nothing is there. So here ATP in the presence of enzyme. Which enzyme? Because here that is activation, it is given activation, so it is called as amino acyl transfer RNA synthase. We can say here amino in the presence of amino acyl transfer RNA synthase, because this enzyme is responsible for the synthesis of transfer RNA and amino acids. That's why it is called synthase enzyme, right? And what happens here? Now here amino acid plus ATP plus enzyme, it forms a complex. We can say here amino acyl because here ATP is there, but this ATP is broken down because without energy, what is the function of ATP? ATP means it provides energy, right? So here energy is consumed, that ATP is broken down, it is converted into AMP, enzyme complex is formed. Amino acyl AMP enzyme complex is formed. That is called activation of amino acid. Now this is the activated amino acids, right? This one is called as activated amino acid, right? Okay, after this activation of amino acids, now second process is charging of transfer RNA. Second, we can say here charging of transfer RNA. In our NCERT, it is also called as amino acylation of transfer RNA. This one is also called as amino acylation of transfer RNA. Another name is there, right? So how transfer RNA means charging of transfer RNA means when this amino acid is attached with transfer RNA, it is called as acylation of amino acids, right? Because transfer RNA, specific transfer RNA are responsible for specific amino acids. We know because they have specific anticodes. According to the anticode, they have specific amino acids. Okay. So what happens here? Because here amino acyl, we can see here amino acyl. What is their product? Our AMP enzyme complex, right? Plus transfer RNA. That is transfer RNA synthesis. Here also transfer RNA synthesis used. Then what happens? Amino acyl tRNA complex here. Now this amino acid is attached with this transfer RNA. What happens? Amino acyl tRNA complex is formed. tRNA complex plus this AMP, this one is released. Enzyme is also released. AMP, it is capital not small. AMP plus enzyme, right? Enzyme is released. Right? Okay, here you understand here AMP is there. We can write here one more plus inorganic phosphorus. Two molecules of inorganic phosphorus are released. Right? They are consumed. Right? It is not arrow means we need not to arrow, but I write here arrow means they are released into the where cytoplasm. Right? And this one. So this what happens here? This amino acyl transfer RNA complex is formed, and this one is called activated transfer RNA. That is charging transfer. This one is called charged transfer RNA. Okay. The second process is there, charging of transfer RNA. Okay. After charging of transfer RNA, then this transfer RNA, it is moving toward the where ribosomes, right? Because it contains specific amino acids, right? So the third process is our translation. This is the third process. Right? After this, what happens? Third process, that is translation process. This one is the actual translation, right? Translation. In translation, this translation is further divided into 3 steps. The 3 steps means what? First step is 3 steps, right? Here, first step is our initiation. Initiation means starting, how it starts, right? Initiation of polypeptide chain. Here, what happens in initiation? First, here we need here, because it is we are discussing about the prokaryotic translation. So we need here subunits. Which one? 50s and 30s subunits. First, right? Second, transfer RNA, that is our, so transfer RNA is also there. GTP or energy, right? Third, for initiation, we need initiation factors IF1, IF2, IF3, right? So these are initiation factor 1, factor 2, factor 3. Some ions we need, like as magnesium, right? Which one? Mg2 we can write here. Mg plus 2, right? So these are the essential things we need here. Okay. These two subunit ribosomes means we can say here 70s ribosomes, GTP, energy for initiation factor is there, magnesium, right? These are essential thing which is essential for this translation. Another things means, and more important we know here, mRNA, I not write here, you can write mRNA and transfer RNA, that is charged, CHARGED, charged transfer RNA. We can say these are the components for translation. We need these things for translation. Okay. First, because these are IF1, IF2 and IF3. These are the initiation factors of bacteria. These were initiation factors of bacteria. It is present in bacteria. So first here question is there, how a small subunit is attached. Small subunit is attached with messenger RNA, right? So small subunit is attached with messenger RNA because small subunit, it has, and the process is attachment of 30s subunit with transfer RNA. Right? Attachment of 30 subunit with transfer RNA, how it attaches? Because in case of prokaryotic cells, right? These means we can say here, that is SD sequence, because mRNA it has code, right? So here in prokaryotic cells, mRNA it contains SD sequence. mRNA it contains SD sequence. What is the full form of SD sequence? SD sequence means shine, dil, garno sequence. This sequence is present. Its SD sequence is present where? In mRNA. And this SD sequence, right? It is this SD sequence, it is made up of, actually it is made up of 8 nitrogen base, and it is also present, right? Before how many? 4 to, we can say here, before 4 to 12 nitrogen base of initiation codon, before 4 to 12 nitrogen base of initiation site or code. So before initiation code, right? before, how much before? 4 to 12 nitrogen base before. A sequence which is made up of 8 nitrogen base is present, and this sequence is called, this group of nitrogen base is called ST sequence, right? So this ST sequence is recognized or identified by just it has anticodes, because we understand here 8 nitrogen base, this is the code which is present in messenger RNA, right? And this 8 nitrogen base code is recognized by or identified by which one? Anti-SD code, ASD code. Another one is ASD, right? Sequence, or we can say code, is present at where? Small subunit. Small subunit means what? We know very well that 70S that means 30S ribosome, it is made up of 16S RNA. It is present at 16S RNA, ribosomal RNA, 16S RNA of smaller subunit. Right? So this messenger RNA, we can say, or this small subunit, it recognizes the SD sequence or ST sequence, recognizes ASD sequence. Because they are complement, due to the complementary, they can recognize, and then it attach with small subunit. Means messenger RNA is attached with the smaller subunit, because that is ST code is present in messenger RNA, right? And just anti-SD code, it is present in 16S ribosomal RNA, because they are complement to each other, so they can bind to each other, right? In case of eukaryotic cells, when we consider the eukaryotic cells, in eukaryotic cells means what? In eukaryotic cell, we know very well there is 7 mg cap, right? Just before the initiation codon is there. So this 7 mg cap, right? They have some specific sequence, and thus opposite specific sequence is present in 40S small subunit. 40S small subunit means in 18S ribosomal RNA.